Thank you very much for the kind introduction. I'm very happy to be here and also like share our perspectives on precision medicine in IBD and chronic inflammatory disorders, because in the last part of the talk, I will also give the outlook on other chronic inflammatory disorders. I want to touch base with you and start with a clinical observation of inflammatory bowel disease. For me as a gastroenterologist, this is my daily bread, but like for many people of you, this is something which might not be familiar with you. So we're talking about Crohn's disease and ulcerative colitis. They are chronic inflammatory disorders which can affect the whole gastrointestinal tract. But what's also important, these are all pictures from patients with IBD. They are basically present as a chronic systemic inflammatory disorder, which can present all sides of the body. And we see a steep rise of incidence and prevalence, especially in the Western countries. If we talk about precision medicine, we very quickly come to the term omics medicine. So we're trying to delineate the complex pathophysiology using different techniques to understand what's really going on. You can also see this by the PubMed entries that if you look at precision medicine or omics, you see that the number of entries are constantly rising, indicating that we need a very complex picture to understand these complex diseases. And if you want to delineate a complex disease, you have to look at from two different dimensions. The first dimension is that you have to look at different compartments of the body to understand what's going on. So not only at the site of inflammation, which is on our perspective is the gastrointestinal tract, but also using all kinds of available biosamples to get a whole picture of the pathophysiology. And the other point is that we need to include many different layers depicted here: the genome, the methylome, the metabolome, the transcriptome, so many different features that try to kind of stack up the topography of this complex disease and then really identify novel pathophysiological pathways. But as a gastroenterologist, I also have to critically ask, what is the unmet clinical need in IBD? But also, what do other disciplines teach us, and how far have we gone so far, and what is currently established in clinical routine here in IBD? And if we look at other disciplines, we see that, for example, with genomics, we see a big impact of genomic information on the grading and also the classification of CNS tumors. This is a paper published in 2018, seeing or indicating that the genomic information is much more valid in the classification as the old established pathological grading. And the basic seeing is so far that we can, like from a gene, we can develop into a therapy where we use a molecular tumor board and identify novel interaction points that can be directly translated into novel therapies. And this is just a picture indicating that in the field of oncology, this is really working. So there are more and more molecular tumor boards established, and the clinical application of multi-omics analysis into the direct precision medicine is something which not only takes place, but also showing in this paper that the application of the molecular tumor board and the stringent follow-up of the recommendations that result from the molecular tumor board also lead to an increased overall survival, indicating or just highlighting again that we need this molecular medicine approach also in other complex diseases as chronic inflammatory diseases or inflammatory bowel disease to really improve the patient care. But how does the clinical reality in IBD currently look like? So if we have a patient with a high disease activity and we make the diagnosis and also the indication for novel drug therapy, we choose a biologic agent, and then we have different trajectories. So either we have a super responder here shown in blue, which directly goes into response, or we have a late responder, or we have the complete non-responder, a little bit what also Dr. Lediger introduced. Upon the failure of the first biologic agent, then we have the second biologic agent or the third biologic agent. So we have a high recycling of biologic agents aiming to kind of target the correct pathophysiological pathway, which then ultimately would lead into the therapy response. The question is, how can we currently improve the IBD therapy in the future? And this is a landmarking overview article from a colleague in Oxford, just like looking at different aspects that might be important in the future. And I want to kind of highlight on the molecular classification, and now will lead you into the host-microbial metabolism, why we think that this is of importance. And the whole story begins like leading many other transcriptome data behind. It's like the whole story starts. We look at a very dense perspective, and at a dense perspective from the technological side, and also from the longitudinal sampling side. This is a study which was done by colleagues at the Broad Institute, where patients with IBD were like biosampled at a very dense sampling scheme. And you can also see, first of all, that they sampled on many biosamples from the host and also from the microbiota, and then also applied different omics technologies to really understand what's going on the pathophysiology of IBD. And overall, or in a nutshell, not guiding you through this busy picture in detail, you can state that IBD manifests as a metabolic dysfunction of the host and the microbiota. So we have very specific metabolic signatures, for example, short-chain fatty acids, but also bile acids, and also here indoles, that are differentially abundant in patients with IBD, and also with relation to the disease severity. And this also brought us to our first analysis, where we assessed the impact of the host microbial metabolism on the induction of therapy response, and using a classical biologic agent, anti-TNF. And in this paper, we basically analyzed whether the microbial metabolism might instruct the host immunity to be more responsive to the infliximab therapy. And what we observed is that the microbiota of patients that do benefit from infliximab therapy, so here indicated as remission, those patients inherit or incorporate a microbiota which is defined by a high metabolic interchange, so a high exchange of metabolites, which also results into increased synthesis of specific byproducts, fermentation byproducts, for example, short-chain fatty acids. Notably, we could also confirm this in other treatment modalities, indicating that this is a very general principle of microbial metabolism that might instruct the host microbiota. These findings are also further corroborated looking at very recent data from a group in Groningen, where they conducted fecal metabolomics from patients with either Crohn's disease, here CD, or ulcerative colitis, so shown here in UC, and you see the black line is the comparison to the healthy controls. And you see overall that there's a bunch of different metabolite classes that are either overabundant or have a lower abundance in the intestinal microbiome of CD and UC patients. And I also just highlighted in red the short-chain fatty acids, which basically confirm a known metabolite class, which has a lower abundance in patients with IBD. What is interesting in this context, and this is something we need to highlight, and also something that we have not completely understand, is that with the increased or decreased, also with a differential abundance of specific metabolites, we also see the expansion of specific pathobiomes that directly associate with the abundance of these metabolites. And this indicates that there might be specific pathobiomes that have a specific metabolism leading to specific byproducts, fermentation products, which either is a starch fermentation or a high protein or a high fiber fermentation. And this association is something that we have to understand in more detail in order to direct targeted therapies either at the metabolic side or at the phage therapy to really restore the microbial metabolism. So if we want to sum up this in a brief picture, we do believe that inflammatory bowel disease is a microbial metabolic dysfunction. And we have several causes that induce this microbial metabolic dysfunction. And I only want to list a few points. Diet might be an extreme important factor, but we also have genetic as a factor that directly impacts our microbiome. And also putting together many, many different factors that we are not able to delineate in detail, but summing them up in the term of exposome. And we need to understand this microbial metabolic dysfunction because this micrometabolic dysfunction has consequences. It has consequences on the individual disease course. It has consequences on therapy response, as I have briefly touched so far. But the major point is that this has consequences on the host metabolism, and it's currently believed that this consequences on the host metabolism is something which we can, on the one hand, capture as a metabolic biomarker, but also it is something that we have to understand in more detail because it might have fundamental impact also on the overall disease course and the chronicity of the disease. And what is really important is that so far we have several metabolite classes that are identified. I highlight tryptophan, short-chain fatty acids, bile acids, and lipids. But you can also see also from the second speaker of this session today, Joseph Tintelnot, you will see that tryptophan metabolism does not only play a role in inflammatory bowel disease, but it appears to be a general principle of host-microbial immunity that might be an actionable target principle. So I want to briefly introduce you to tryptophan metabolism because in the second part of my talk, I want to dwell a bit on the tryptophan metabolism and also end with some data from our own group. So tryptophan metabolism tryptophan is an essential amino acid, so it provides basically building blocks for NAD synthesis in the cell, and this building block via NAD synthesis is generated via the degradation of tryptophan along the kynurenine pathway. And in addition to the kynurenine pathway, there are two other degradation pathways present, which is on the one hand, the degradation along the serotonin pathway and also the degradation, which is mostly conducted in the microbiota into the indole pathway. It is important that this tryptophan metabolism has really pleiotropic effects on host immunity. We know that kynurenine and its derivatives have a direct impact on host immune response. We know that AHR ligands, which are generated from the microbiota, have a direct impact on cellular immunity, including the induction of IL-22, which is a barrier protective cytokine. And we have, apart from that, we have also the impact of the tryptophan along the 5-hydroxytryptophan pathway, which affects gut motility and gut-brain signaling. So what is the connection between tryptophan metabolism and IBD? Where does it come from? One of the very, very earliest observations was that the tryptophan basically has to be taken up into the cell by a co-receptor. And this co-receptor consists of ACE2 and the BO81. So BO81 is the main receptor recruiting tryptophan into the intracellular lumen, but the regulation of this receptor the expression of this receptor is regulated by a co-receptor which is called ACE2. And it was very previously shown now, almost 10 years ago, that the disruption of this ACE2 receptor leads to an increased experimental colitis, which is a very simple model to study the impact on intestinal inflammation. And what has been shown is that this ACE2 receptor leads to a decreased expression of BO81, a decreased bioavailability of tryptophan, and that this tryptophan deficiency directly impairs the mucosal production of antimicrobial peptide. These were preliminary findings that prompted us to study tryptophan metabolism in more detail in inflammatory bowel disease. We could show in a cross-sectional study that patients with increased disease activity. So you see the control patients, inactive, active, inactive, active for CV and UC. You see overall an increased tryptophan degradation, and this tryptophan degradation appears to happen along the kynurenine tryptophan axis, as you see an increased ratio of kynurenine to tryptophan. It has been also previously shown that the metabolic rewiring of tryptophan metabolism occurs at several steps of this tryptophan degradation pathway, as, for example, the decreased bioavailability of two metabolites, kynurenic acid and xanthorenic acid, which are both generated by this AADAT enzyme, lead to increased disease severity in IBD. So if you want to conclude, one can say that we have this increased tryptophan degradation along the kynurenine axis, meaning that IBD is basically an immunometabolic disease where we have a false degradation of an essential amino acid along the false pathway. And in the last couple of minutes, I want to show you some data from our own group where we basically asked the question whether this tryptophan degradation that we have observed in IBD, whether this is a metabolic principle which is specifically confined to inflammatory bowel disease, or whether this is a more unifying metabolic principle that also accounts for other chronic inflammatory diseases. This is a study which has been mainly driven by a postdoc in my lab, Danielle Harris. And what she did is that we took advantage of our competence center for inflammation medicine, where we basically see a great variety of patients with chronic inflammatory diseases, mainly IBD on the one hand, and also patients with chronic inflammatory skin diseases and chronic inflammatory diseases of the musculoskeletal system, such as rheumatoid arthritis, systemic lupus erythematosus, axial spondyloarthritis. What we had was the routine laboratory, and we included the serum tryptophan measurement into our routine laboratory, so as a standard clinical marker, and we conducted an export of our electronic e-health record, and taken together, we extracted the data from nearly 2,000 patients, which were observed over a time frame of nearly 10,000 of 10 years, summing up the number of clinical observations to roughly 30,000 observations. And for each individual time point, we had the electronic health records and also the serum tryptophan measurements. And we wanted to ask the question whether this repetitive measurement of serum tryptophan might also help us to kind of anticipate the individual disease course of a patient, for which we know that an unrestricted inflammation will gradually lead to complications, to surgery, or to the switch to other biologic therapies. So at the first glance, we wanted to know whether tryptophan degradation is a feature that is also seen in other chronic inflammatory diseases. And if you take all patients with rheumatoid diseases together, you also see a significant reduction of the serum tryptophan in patients with rheumatoid diseases. You can also directly correlate the serum tryptophan values with the CRP values as a generalizable marker of immune activation. And you can also see with varying degree that we see a very strong association of serum tryptophan degradation with disease activity. Next, we wanted to ask whether the tryptophan stability might be an indicator of disease course in IBD. So on the left hand, you see a representative screenshot of the available tryptophan data of a patient, and you see a very, very high fluctuation over time. So what we did is that we looked at this fluctuation over time, and we asked a very simple question. This was, do patients over time will require a biologic therapy? And biologic therapy is the first treatment escalation that every patient will undergo if the disease is not under fully control. So the term treatment escalation, which is here defined as a biologic status in change. So patients either never received a biologic therapy, or they had to be changed onto a therapeutic therapy, or they have always been on a biologic therapy, indicating an even more severe disease course. And we associated the standard deviation of this tryptophan. We observed that indeed those patients which have a more complicated disease course also present with a higher fluctuation of the tryptophan, indicating that tryptophan stability, metabolic stability of the tryptophan metabolism is a marker that indicates individual disease complications. Then lastly, we wanted to understand what defines a patient with high or low tryptophan metabolism. So we again conducted a multi-observation cross-sectional design study where we recruited patients, either healthy control patients with a Gaussian with a Gaussian number of tryptophan values, and we specifically selected patients with either high or low tryptophan values to understand the extremes. And this is how the patients look like. And we biosampled those patients, and then we conducted a targeted metabolomic screen where we used the biocrates assay. And first of all, we basically looked at the marker of tryptophan degradation. So looking at the kynurenine level in IBD, rheumatoid disease, or skin diseases. And we also calculated the kynurenine tryptophan ratio, indicating that those patients that show a decreased serum tryptophan concentration are indeed having an increased degradation of tryptophan along the kynurenine axis. So it is an active enzymatic degradation that we observe across all chronic inflammatory diseases. And lastly, we also look for the accumulation of indoles, and we specifically look for indoles that are known to be HR inducers. And we also look for indoles that appear to be not HR inducers. And what we observed, which is not shown in this panel here, that the majority of indoles that are HR inducers were decreased in patients with low tryptophan, but we also observed one very interesting accumulation of indoxyl sulfate, which is a indole which is further metabolized in the liver and is basically known as a uremic toxin. And this indoxyl sulfate was one of the indoles that were specifically overabundant in patients with tryptophan low. So to sum up, I hope I have kind of convinced you that chronic inflammation is characterized by a host and microbial metabolic dysfunction. And so far, there are only very few metabolic principles that are identified in IBD. So we're only looking at the tip of the iceberg. We have established that tryptophan degradation is a unifying metabolic feature of chronic inflammation. And based on this, we have to now test in more translational trials whether we can use therapeutic interceptions along this tryptophan degradation pathway as a novel therapeutic interaction point. With this, I want to give credit specifically to the people in my lab and also to the funding agencies and the collaboration partners, and I'm happy to take questions.
Thank you very much, Professor Aden. I love that you addressed other inflammatory indications as well, and not just inflammatory bowel diseases. We talk a lot about commonalities of diseases as well. So that was great to see. I think we have time for one or two questions. There was one question about how to measure tryptophan in a routine diagnostic, but maybe we can open up the question a little bit and talk about what's already possible with routine diagnostic methods and how you see omics developing into to something that is used in clinical routine to guide therapeutic decisions.
Yeah, very interesting and important question. So the first part of the question, we are able to measure tryptophan in our clinical laboratory using HPLC. But I think the most important point of this is basically open up to omics analysis is that we have to work on the turnaround time, because ultimately we have one benchmark, and this is CRP, and we know about the ups and downs of the CRP. But the big advantage of the CRP that this is a marker which is immediately available, so within 1.5 hours. And if we want to go for if you want to go for something that will really help us to guide a therapy, we need at least a turnaround time of something like between 2 or 3 days, because in many clinical indications you might need to make an urgent decision. So in order to translate this into clinical medicine, we really have to work on speed and the availability of omics data. This is something that we also understand, that we're also currently working on. And but I think we will not be the only one which are tackled by this problem or this unmet clinical need.
Okay, thank you very much.